fatal, but can be avoided once appropriately vaccinated. So this is an initiative from Cadila Pharma through its innovation and to serve the society and humankind. The topic is today's webinar is novel 3-dose G protein vaccine, which is ultimate key of compliance. So, dear Dr. Travis, the world's first 3-dose novel vaccine, and with great pride, we would like to inform you that Travis has successfully completed of 1 year of his launch with more than 40K animal victims being vaccinated. 100% compliance was recorded, and no case of ADRs was registered, sir. So with this, sir, it's my honor and privilege to introduce our today's speaker, which sir's name is enough, Dr. Durga Madhav Satpathy, HOD, MKCG Medical College. So, sir, now I would like to request you, sir, to Dr. Durga Madhav Satpathy, sir, to initiate the talks, and later on we'll have discussion of question and answer session. All participants can drop their messages, questions in the drop box, and we'll read all out the question after this presentation. So thank you, sir. Once again, sir, requesting you kindly have your own talk, sir, regarding this, sir. Thank you so much, sir. I'm handing over to you, sir.
Thank you, Satya. Yes, good evening, everyone. And yes, as Satya said, that today we are going to discuss on an important zoonotic disease, but ancient in nature, that is rabies, which is, as we all know, that it is 100% fatal in nature. Once an unfortunate person is diagnosed with clinical rabies and it is written on the prescription, then it is equivalent to writing the death certificate of that poor fellow. So we know that yes, rabies is 100% fatal. However, it is preventable in nature. We can be able to prevent it. But still, though ancient in nature, and in fact, the word rabies comes from the our own Sanskrit. It is described in the Rig Veda. The word comes from ravas. Ravas means to act abnormally or furiously. Though it comes from the ancient times, but still, unfortunately, nearly 60,000 deaths occurs in the world, of which nearly 20,000 of human deaths due to rabies occurs in our country. That is the most important fact which has to be taken up. So these deaths can be prevented, and 91% of deaths, these deaths occurs in the rural places of our country, and this is one of the worst diseases to have afflicted mankind. And we can see that nearly every 9 minutes, a person in our country dies due to this zoonotic disease, which is 100% fatal, that is rabies. Next, please. Though, as I told you that, yes, this is 100% fatal in nature, but it can be prevented. And this prevention is most important. And this preventive part of animal bites, which leads to rabies, stands on the tripod of three things. That is the local owned treatment. Second, to generate antibodies within by our own immune system through active immunization, which is the process of vaccination. And the third most important is the use of passive immunization, that is immunoglobulins, either immunoglobulins or monoclonal antibodies. We have immunoglobulins are of two types. Either it can be equine source ERIG or human source HRIG, or it can be a monoclonal antibody against rabies, which can be single monoclonal or a mixture of two monoclonal antibodies. So this passive immunization is definitely has to be also taken up, especially in category 3 exposures. I want to highlight at this point that any animal bite leading, which is transdermal in nature, that is leading to bleeding or oozing of blood, oozing of a single drop or severe bleeding, whatever it is, if there is a oozing of blood or there is bleeding from the wound, it becomes category 3. And in this, apart from active immunization, we have to also practice passive immunization, that is the use of either immunoglobulins or monoclonal antibodies against rabies. Coming to the vaccination part, the vaccines which are used at present in the intramuscular route are on the days of 0, 3, 7, 14, and 28, in which the day 14 is omitted if it is an intradermal rabies vaccine, but if it is an intramuscular or SN regime, then 0, 3, 7, 14, and 28 are the days. And one thing I want to bring forward at this point is that the day of the first vaccination is considered to be day 0. Ideally, this day 0 should be on the day of bite. That is, on the day of bite, we should also give the vaccine. But unfortunately, we get cases even after 1 or 2 days or 4 days after the bite. But whenever we give the first dose of the vaccine, that is considered to be the day 0. And from that date, we have to calculate the other days, day 3, day 7, day 14, or day 28. So this is what I want to highlight. Ideally, the day of bite should be day 0. But whenever you are giving the vaccine, first dose of vaccine, if it is on the day of bite, it is day 0. If after 2 days also, that is the day 0. So from that day, you have to calculate the other days. That is day 3, day 7, day 14, and day 28. Next, please. These 5-dose vaccination schedule which is being given, especially when you are using the cell culture vaccines which are used currently. One of the major things which we find is that they are a bit less compliant. That is, day 037, within a week you complete 3 doses. The next dose comes after 7 days. And the last dose comes after 14 days of this, or after 3 weeks of the day 7. So at present, sometimes we face non-compliance. They usually take 037, but fail to forget or fail to come or forget about the day 14 or day 28, which is after a month. So this is one of the important things which has to be taken care of. And though stability is there, adverse events are also not that high in cell culture vaccines, but availability at times becomes short because the huge number of cases who require post-exposure treatment, we cannot expect that we can be able to produce enough of vaccines for this SN regimen or intramuscular regimen. So that is at times a problem, but the main thing is regarding compliance, where many a times patients forget about day 14 or even day 28. They come after that. So this is one of the important things as far as the cell culture vaccines used in post-exposure prophylaxis is concerned at present. Next, please. So compliance, as we know, for those who have received the full course of ARV, anti-rabies vaccine, delayed compliant who completed the scheduled dose of ARV within 60 days but deviated, and dropout who had taken at least 1 dose or 2 doses and they did not complete the rest of the schedule. That is what we mean. But we want that for a disease against 100% fatal disease against rabies, all the vaccinations should be compliant for the anti-rabies vaccination schedule. We do not want either delayed or dropout is not at all desired. We want everyone to be compliant in nature. So that is one of the most important points as far as active immunization against rabies is concerned. Next, please. So, in view of this, analysis was done and it was found out that patients do not always receive optimal rabies post-exposure under real life conditions. Some drop out, and because of this dropout, there is a lack of adequate immunostimulation, thereby leading to a decreased active immunization response by the body. So these are some of the complications which can occur. If even 4 doses of the vaccine are given, 0, 3, 7, 14 also, nearly 7% of the patients do not develop adequate antibodies, seroprotective antibodies, which should be more than 0.5 international units per ml. So this is one of the studies they have found out as far as the 0, 3, 7, 14 and 28, there is 5 doses of SN regimen is concerned. Next, please. And these are some of the cases which has been well documented that even after 3 doses, some have succumbed. Of course, in majority of these cases, you can find out of this case series that the main drawback was one, only 3 doses had been given, and the next most important thing is the lack of use of passive immunization. I told you that in the first place, the tripod of anti-rabies vaccination or rabies prophylaxis, post-exposure prophylaxis against rabies stands on 3 things. The first, local care of the wound. Second is passive immunization. The third is active immunization. Because we know that active immunization is a process. The antigen has to be administered. It needs to do a thorough immunostimulation by which not only development of anti-rabies antibodies, that is seroconversion, but we want seroprotective levels of antibodies. That is seroprotective levels of antibodies, which is more than 0.5 international units of antibodies per ml. So for this, the necessity of use of passive immunization, that is either immunoglobulins, ERIG or HRIG, or the use of monoclonal antibodies in all category 3 cases is a must. So you can see in this case series, even after 3 doses of vaccine and due to non-use of passive immunization, they have succumbed. The clinical features may differ than a classical case, but or the length of survival can be a bit more in case of partially immunized or partially treated cases. But however, the non-compliant vaccinees and wherever passive immunization has not been done, especially in category 3 cases, then we are putting the cases under risk for which they can face the 100% fatal disease of rabies. So this is what I want to highlight, that we have to go for completing the entire course of the vaccine as well as the use of passive immunization for category 3 cases to prevent such a fatal diseases as rabies. This is what I want to highlight. Next, please. So, as I say, a hospital-based study wherein 53% were non-compliant to the full course of ARV, which has been documented by a study conducted at our own country at Safdarjung Hospital, New Delhi. So, this is one of the important things, that we do not want anyone to be either non-compliant or delayed compliant. We want everyone who has been afflicted with animal bite and has been receiving post-exposure anti-rabies vaccine should have 100% compliance to the full course of the vaccination. This is what I want to highlight here, because studies have said that at times 0, 3, 7, 14, and 28 also has some non-compliance. Next, please. So compliance of anti-rabies vaccine, this is what has been found out at Municipality Corporation at Bangalore also, wherein 215 animal bite patients were treated, and 0, 3, 7, 14, and 28 were the intramuscular doses, and also measures constraints for compliance to complete the course was seen, wherein from 100%, the full course of vaccine was taken by only 60% of the patients. So the compliance rate for intramuscular rabies vaccination was found to be 60% in another study conducted at Bangalore, wherein SN regime, that is 0, 3, 7, 14, and 28 was used. Next, please. And the causes, various causes for not completing this whole 5 doses or 4 doses, whatever, if it is IDRV, the major cause was by the cases as loss of wages, because they have to come from a distant place to take a vaccine, and by that they lose a single day, and once they lose a single day, they lose the wage for that day. So that was the main cause. The other causes like school timing, children usually go from 10 a.m. to 4 p.m. to the school, and by 4:30 when they come, the clinic is closed. Likewise, distance to the hospital may be very high, and also forgetfulness, especially day 14 and day 28, which are a bit far from the original dose. These are some of the causes which were found out, especially for the non-compliance to the SN regimen of anti-rabies vaccination. Next, please. So for this, let us take up how anti-rabies vaccine evolved in the first place. In the first place, as we all know, that anti-rabies vaccine, the first anti-rabies vaccine was the nerve tissue vaccine, NTV. And this vaccine had been developed since long, when the famous Louis Pasteur, Sir Louis Pasteur, administered the first rabies vaccine to a boy named Joseph Meister. That was a nerve tissue vaccine. And this was used for a long time, till 2004, even in our country. After 2004, our country also discontinued the use of nerve tissue vaccine. The reasons being that less potent, for which we have to give it on a daily basis, and the volume of the vaccine was also very high, 5 ml or 3 ml given subcutaneously on a daily dose basis for 10 days or for 12 days for class 3 bites. So from 2004, and another important thing is that as it was derived from sheep brain, how much of filtration, purification occurred for manufacturing this vaccine? Some amount of myelin tissue of the sheep still remained in the vaccine, and there occurred a cross reaction within this myelin of the vaccine to the myelin sheath of the human nervous system, leading to the fatal neuroparalytic reaction. So from 2004, this nerve tissue vaccine, the use of nerve tissue vaccine has been stopped in our country. Then came the cell culture vaccines. Cell culture vaccines, anyway, they were there in the country from 1911. They were of various types, and this cell culture vaccine were usually inactivated in nature, and they were from especially 3 cell culture sources. One is the human diploid cell, the second purified chick embryoblast cell, and the third is the purified Vero cell. These are the 3 cell culture vaccines which were used as an inactivated vaccine for intramuscular dose, and we have got it. These vaccines are safe and potent, for which we do not need daily doses, and the dose also decreased maybe from 0.5 ml to 1 ml, depending on the volume of diluent of that vaccine. And it was given on not a daily basis, but on 0, 3, 7, 14, and 28, as I had discussed earlier. And if these vaccines are given on as the intradermal route, also the dose is minimum 0.1 ml on two sides, given on days 0, 3, 7 and 28, day 14 being omitted. So this is how the rabies vaccines evolved from nerve tissue vaccine to the cell culture vaccines at present. Next, please. Next, please. So for this, because of this non-compliant nature, and 5 doses have to be taken on various days with a long gap after the first week, another week, then another 2 weeks. There occurred a change for another formation of a vaccine. This was based on the fact that, as we all know, and rabies virus is a bullet-shaped vaccine, and the main antigen against which, if we develop antibodies, is the G protein present on the capsular on the viral capsule. The G proteins which are present, which are the spike proteins which are present, which is termed as the G protein. And if antibody is developed towards this G protein, then it is able to destroy the virus. So this, therefore, only the G protein was taken for developing the anti-rabies vaccine. So this is the first protein subunit vaccine against rabies. You know, this subunit vaccines are the concept of development of subunit vaccines are not new. We had hepatitis B since more than 3 decades by now. That is a subunit vaccine against hepatitis B virus. Likewise, influenza vaccines are also subunit in nature. So subunit vaccines are not new, but this was the first protein subunit vaccines developed against rabies. And this is prepared on the VLP platform technology. VLP means virus-like particle. This is based on preparation of this technology of VLP platform technology. And because this vaccine only has the G protein in a concentrated form, so 3 doses of this vaccine is sufficient to generate seroprotective level of antibodies. And as 3 doses of vaccines are to be given, therefore the compliance becomes better, and it was convenient. And the main thing is, because it is not a cell culture vaccine, which is an inactivated vaccine, wherein it has the inactivated viruses which has been grown on a particular cell culture and has been inactivated and freeze-dried. So it is termed as a lyophilized vaccine, for which it has to be again diluted with this diluent before the use of the vaccination for active immunization. But as this is a subunit vaccine, so no need to lyophilize it. It is available in a liquid form by which we can directly simply draw the vaccine from the vial and inject it. So no need for dilution. So these are the new technologies which came out for this novel 3-dose G protein vaccine. Next, please. Next. As you can see here, that the previous one, yeah. From the left corner, bottom corner, we can see the entire rabies vaccine, the bullet-shaped vaccine. A cross-section has been given here. And this outer capsule contains the G protein. And these proteins are used for manufacturing the VLPs or virus-like particles similar to the rabies virus particles, but it does not contain the genetic material or the genome. So for that, all the VLP vaccines, they preserve the characteristics of the virus, but they do not contain the genome of the virus. So it becomes a concentrated source of G proteins only. We do not have the full virus. The G proteins are there, which is essential for immunostimulation and manufacturing of antibodies targeted towards this G protein by our immune system. This is the difference from which you can see from the left bottom to the right hand top, that it is similar in nature. However, you can mark that this VLP technology, by this, the structure is maintained, but the genome is absent. So we get a, by this VLP technology, we have got a similar structure as with a concentrated source of G protein. Next, please. And this vaccine has been used. Trials were done because it was a collaborative study between US and our country. And this was used with 10 microgram on 037, 10 microgram 03, 50 microgram 03, and 50 microgram 037. And we can see that in this, the advantage is wherever we used this 50 microgram of this VLP technology based vaccine, and used on 037 and compared with the normal SN regime, the antibody titer was quite similar to the others. So this is one of the important findings of having the titration of this VLP-based rabies vaccine to contain 50 microgram of this vaccine and to be used on days 0, 3, and 7. If that is done, this vaccine is proven to be efficacious. Next, please. And after that, clinical studies were done with using this 50 microgram of VLP technology-based novel G protein vaccine, and it was compared with a one of the leading cell culture vaccines, that is the purified thick embryo fibroblast vaccine, and it was compared. In one arm, the group of patients took the purified thick embryo fibroblast vaccine on 037, 14, and 28, and in the study group, they took it 037 or 50 microgram of G protein vaccine. And this study has been even documented by the researchers, and it was found, it was especially seen for the safety as well as immunogenicity in a comparative view. Next, please. So this is how they went. 533 subjects were given the 037 novel G protein vaccine. The rest 267 were given 037, 14, and 28 as per the prescribed dose of that vaccine, the purified thick embryo fibroblast cell culture vaccine, and they were followed up for nearly 1 year. Next, please. And we can see whenever we are going for any trial of a vaccine, two things have to be taken up. Two things has to be considered, otherwise we fail in our nature. One is efficacy. That is it efficacious in producing adequate seroprotective antibodies as per the comparator? The second is it also safe or not as per the comparator? So this efficacy and safety both have to be taken up. Otherwise, we cannot go for the use of a vaccine, especially a new vaccine. And here we can see that as far as efficacy is concerned, quite safe. The results are consistent against different age groups with no significant variation in the amount of antibodies generated. So it shows that to be non-inferior in as far as seroprotection is concerned for the both primary endpoint at day 14 and day 42. As regards the test vaccine is concerned. So that shows the efficacy of this vaccine. The second comes the safety. And as far as safety is concerned, again, we can see the reactogenicity is also similar or rather less as compared to the corresponding comparator vaccine, though none of them had serious adverse events, SAEs, but again, either the local or some systemic like fever, headache, malaise, they were quite similar, rather somewhat higher in the comparator vaccine as compared to the G protein vaccine, because this G protein vaccine only contains the protein. There is no other components of the virus. So definitely this the side effects, especially the safety, was also proved to be better in this G protein vaccine. Next, please. Next, please. So this conclusion is that it is definitely non-inferior to the reference vaccine, less adverse events, and simultaneously was found to be safe as well as immunogenic with the comparator to 5 doses of WHO pre-qualified vaccine. So both as far as efficacy and safety is concerned, this novel G protein vaccine, which was used on 037, was found to be safe as well as efficacious. Rather, it was found to be efficacious and safe as per the comparator vaccine. Next, please. And for this, because these vaccines have been used and compliance is better, even the company also has received many awards, the Golden Globe Tigers Award or the award from the Economic Times, that is Pharma Innovation of the Year, because this is the first subunit G protein vaccine against rabies. And with 3 doses of the vaccine given on 037 without reconstitution, because it is a liquid vaccine, it was found to be both efficacious as well as safe. The awards also went to the manufacturers for this. So this is what I want to highlight, all the respected caregivers, especially whenever animal bites are concerned, that whenever we get a case of animal bite, if it is category 3, wherein I want to again reiterate the fact that category 3 means bleeding from the site of bite. It doesn't mean gushing out of blood. Even a single oozing of blood is also category 3. Gushing out of blood is also category 3. That means the bite is intradermal, transdermal in nature. And any transdermal bite is taken as category 3. And here, apart from local wound washing, thorough washing with soap and water for 10 minutes, the other is to use the passive immunization, and the next is to go for active immunization. Whenever we are using active immunization, we have to be ensured that the full course of this anti-rabies vaccine has been taken by the client, because this being a 100% fatal zoonotic disease. And for this, as far as compliance is concerned, this novel G protein vaccine, wherein only 3 doses, 037, only 3 doses, and within a week, the entire anti-rabies vaccination course becomes complete. So you can be having a safe as well as efficacious vaccine in your hand to fight against this 100% fatal disease. Next, please. Thank you, everyone, for your patient hearing. In fact, I hope that the message which I have given, that rabies is 100% fatal, the only way is that we have to prevent it. And the prevention stands on the tripod of 3 things. 1, local care of the wound. Second, the use of passive immunization, especially for category 3 exposures. And I want to highlight category 3 means any transdermal exposure, wherein there has occurred bleeding. Even oozing of blood or gushing out of blood, whatever it is, if it is a transdermal exposure, where there is oozing of blood or gushing of blood, it is a category 3 exposure. We have to use passive immunization either in the form of immunoglobulins or in the form of monoclonal antibodies against rabies. And the third is vaccination, active immunization. And as far as active immunization is concerned, now the only thing which we had till now is the use of cell culture vaccines. But whenever we are going for active immunization, we have to ensure that all the doses have been taken. All the doses have been taken for SN regime or cell culture vaccine. It is 0, 3, 7, 14, and 28. Whereas here, now we have another VLP platform based G protein vaccine, which due to its high concentration of G protein, found to be efficacious and safe. Only 50 micrograms of it is needed, which is present in 0.5 ml to be given intramuscularly, can be given on 3 days only, 0, 3, 7. So within a week, the entire course of active immunization is completed. So we can be ensured of total compliance against an active immunization. So this is what I want to bring to the August gathering here, August and eminent gathering. And thank you once again for your patient hearing. Namaskar.
Sir, thank you so much, sir. Really now 79 delegates are there, sir, online, which I can see 79, sir. From head office, I came to know, sir. So thank you so much, sir, for this informative talks. And I would truly appreciate your efforts for this, sir. As you said, sir, last within one year only, sir, Travis has awarded two very good awards, sir. That is ET Pharma Award and also Golden Globe Tiger Award, sir. So one more thing, just I would like to inform to all our delegates that when trial was going on, sir, Dr. Durga Madhav Satpathy, Dr. Durga sir was the part of this trial, sir. Also, he was in Sambalpur that time at Burla, sir. So thank you so much, sir, for taking care of this Travis, sir, special, sir, for supporting us, sir, for motivating us, sir, further innovative products and to move further, sir. Thank you so much, sir, for this session, sir. We will always.
Because patients for therapies are waiting outside.
Okay, okay, sir. The next person will take Travis, sir. Thank you so much, sir. Thank you, sir.
Doctor,
Siddharth, from your end. Siddharth.
am I audible? Am I
Siddharth,
audible, sir?
Yes, Siddharth. Please, sir, yes.
Sir, there are some questions on the chat box. Can you, Satya sir, can you please read them for the doctor?
1 minute.
Can you one thing?
Suppose
Satya,
if the patient is get bitten by
you please note it down, and we can you can send it to me. I will give you in writing, and you can revert back to the audience. Okay.
Okay, done, sir.
Okay, sir. Okay, sir. No issue,
If
sir. Thank you so much, sir.
Right.
Sir, thank you so much for 1 second, sir, from entire Cadila team, sir, product head office, and from field also, sir. Thank you so much, sir.
Thank you, sir. Thank you.
Hello, sir. Hello.